That was impossible to fit for us.
Those are astounding findings.
That is really impressive.
Have you shared these with colleagues?
Yeah. It was really impressive. They dropped from their seats, actually. We did, in the sampling, we used what is called gel electrophoresis. And it's a technique that we use for sampling genetical material and chromosomes. They have the genetical material. And at this time, the only thing that we could find that it was closely resembling those 9 chromosomes were dolphins only. Dolphins.
Doctor, I'm just a layman, so I'm going to ask a layman-like question on behalf of the audience. As you well know, people can tamper with photographs on computers and fake them.
Yes.
Is there any way that you can see that anybody easily could have tampered with something at the molecular chromosome level to have produced this as a hoax?
Okay. In terms of the technology that humanity has right now,
yes,
we can deal with genes. Yeah, we can open DNA strands, but as you know, even the human genome is not completely set right now. So we don't even know our own DNA system completely. We cannot say actually how many genes we have working for certain situations in our body. So it's very difficult right now to tamper something like this. It will be very difficult to add a chromosome to a cell and keep it alive. And especially a chromosome, because you have to link them all together. Chromosomes, they react to specific metabolic reactions in the body. So for right now, I would say that for humanity, it's impossible.
All right.
No way.
Then that must mean that, in your opinion, you examined tissue and muscle and blood, what we're calling blood, of a being not human and not of any mammal construction.
Not human or any mammal that we have seen or we have ever cataloged.
Art, ask him about the other tissue samples.
You ask him
more, not just.
No, you go ahead. You ask him. What other?
Cohn?
Okay. Yeah.
Talk about the cranial tissue.
Yeah, that is very interesting too. This is the other findings that I'm going. In the samples that Dr. Reyes gave me, there was apparently he put the samples from 1 to 4, but in fact there were 5. One of the vials actually contained 2 tissue samples stuck together due to their growth relationship. It's normally that they grow together. So when Dr. Reyes took this sample thinking that it's just a single thing, he took 2, actually. That second sample consists of what it would be brain matter. In this case, that would be the third sample. It resembles what is called the dura and the pia matter of the human brain. It resembles them. This tissue is a very fine vascular membrane which cushions the brain against the skull. It would be like the folding. It would be like a bag that the brain is actually floating inside this bag. And it protects it against the direct contact with the skull bone. This lining, we should call, extends over the entire surface of the gray matter of the brain. So this is covering the whole brain. In this case, was a dense, unelastic, and fibrous material lining the skull inside. Below this tissue, we found what is called the subarachnoid space tissue, and it's like a spongy or a gel-like tissue, consisting of a delicate connective fibrous material, meshy. In humans, it's called the subarachnoid fluid. We found this to be a similar material, but unlike in humans, it contains large, really large nerve endings and oval or sometimes round surfaces. These nerves are totally different and foreign to anything we even closely relating to the human nerve tissue and our anatomy. This is completely different. The arrangement is completely different on nerves. Another really interesting finding is that under closer examination, this tissue area did not at all resemble human tissue. we more closely study, the cranial membrane resembles probably the one of a reptilian type animal, because it have a much different structural anatomy.
All right, Doctor, we're very short on time. You've given us a very great deal of information. Is there anything else critical to get out before we move on?
Yes, it's something really, really interesting. We found some new structures inside the brain.
New structures?
New structures inside this. The only part of the brain, you know that the brain's got certain convolutions. When you look at a brain,
sure.
The only part that we have from this sample was what it would be considered the external or the molecular layer. Okay, the fascinating and unique differences found in this sample was the discovery of some angular and tubular straw-like open membranes, like little connecting tubes, that they are completely wrapped in nerves and connecting fibers. These little tubes are protruding up and through the base of the molecular layer. So they're coming out of this layer in the brain, I should say the brain cells. They're randomly looping over the top of the cortex, only to descend back into the surface, that'll be the convolution surface, and they're embedding directly into the cells of the body of Purkinje. The Purkinje cells are the ones that are related to the electrical capacity in the human brain, and they work as an isolation material. These cells are actually attached to the axon of the dendrites. These are nerve cells. But in this case, there was a different structure that we don't know. We haven't seen, never seen this connection before. This is truly a new discovery.